Hi, everyone. Thank you so much for coming today. Like Aitan said, during my time at the Advanced Bioimaging Facility, I was a microscope specialist, and I was meeting with researchers just like you. I would be discussing with them what their project was, what they wanted to see, what they were hoping to find, and based on that information, we would discuss what kind of fluorescent probes they needed to use, as well as what type of microscopy application would be best suited for their research question. Then I would train them how to use those systems and then how to do the analysis after the fact. So generally the way that these kinds of presentations work is you start with the most basic form of microscopy and you keep adding layers of complexity until you get to the most advanced types of microscopy, but that can be a bit difficult to translate into how to apply that technology to your sample. So I'm doing it in reverse this time, where I'm going to focus on general common types of samples that people use in research and the best type of microscopy application for that particular sample and what you want to find. So we're going to start with whole organisms, the largest kind you can use, and we're going to zoom in, pardon the pun, to organs, tissues, single cells, and single molecules. But before we get into that, just in case, I want to make sure that everyone's on the same playing field, and I just give a general overview of how fluorescence works. So on the left here, we have a fluorescent molecule. In this case, it's DAPI. It's a DNA binding molecule, and
can be excited by the absorption of light. Can be excited by the absorption of light.
Sorry, I don't know why that was there. It's embarrassing. So you have a particular wavelength come in and excite that fluorescent molecule. And in this case, it's a UV light. And all of the electrons in that fluorophore are going to come to an excited state. And while the electrons are in that excited state, they are going to lose some of that energy due to vibrations and other factors. And then all of the energy will be released at once as light energy. But due to that loss of energy, that wavelength is now longer and has lower energy. So on the right here, we see the excitation curve of DAPI. Light within this range can excite that molecule. And then we have the emission curve of DAPI, where that light will be emitted as a longer wavelength. But typically speaking, people don't want to just see one particular structure in their sample. They want to see multiple structures. So using the excitation curves and the emission curve information of the different fluorophores that we use, we can overlap multiple dyes to look at multiple structures together.
Can be excited by the
Goodness, I'm so sorry. Those are supposed to be gone. And then just to go over how a basic microscope works, we have a particular filter that's going to single out a particular wavelength of light. In this case, we have blue light being filtered out from the regular white light. And this sample has a green fluorescent molecule. So being excited by blue light, it will emit a green fluorescence that will pass through the mirror, through the green filter, and then to the eyepiece where we can visualize it. So with that underway, we can talk about the different kinds of samples that we have. So in this example, we can look at whole organisms. And the way that people tend to look at whole organisms in microscopy is using a technique called light sheet microscopy. Light sheet microscopy is actually really old, but we weren't able to use it because the technology wasn't advanced enough for us to image enough things. So it's a little bit different from traditional microscopy, where you have the excitation light is actually delivered to the sample in a beam or a very thin sheet, therefore the name light sheet. And then the emission, the fluorescence, is actually collected perpendicular to the sample. And this particular setup allows us to have very fast optical sectioning, which means that we're able to image the sample in 3 dimensions very quickly. We're also able to manipulate the sample. In some microscopes, you can actually rotate the sample in 3 dimensions to get the optimal imaging view of your sample. And we are able to, for example, image neuronal activity. This is a zebrafish larva. And if I play the video, you can see little flashes of light. And every time you see a flashing light, that's actually a neuron that's being excited because it has a calcium channel labeled fluorescent protein. So we have our resolution in XY. but then we also have a depth of the image as well. So we have a three-dimensional representation of our sample. If you want to image whole organisms, it's really important that the sample is transparent. So these are larvae before you have too many lipids that are settled in that are going to scatter that light. One of the advantages of light sheet microscopy is that you can image very large samples. For example, the larva of a zebrafish is several millimeters, but usually traditional microscopy can image about 100 micron thick samples, and it's able to do this very quickly. One of the other things that people might use light sheet microscopy for is for just studying development. So this is an example provided by Zeiss of a Drosophila embryo going through development, taking place over 11 hours. So they imaged this in 3 dimensions through time, and able to do that very quickly as well. However, if you don't study development and you don't have access to transparent samples, and you want to look at, say, whole brains, hearts, lungs, that kind of thing, then you end up having a problem because those organs are very opaque. And they're opaque because they have lipids, and lipids scatter light. So there were new techniques that were developed called clearing, where essentially you remove the lipids from that particular organ using specific chemical solutions. And there have been a lot of new protocols developed. So the first one was Clarity in 2013, and then you had things like Switch and Shield. I'm not going to get into the specifics of how each of those work. I'll just provide one example, but if that's something that interests you, I highly recommend that you go look at the Chung Lab and their resources, where they have all of these different protocols. And the good thing is that the longer that these protocols and these things are around, the more accessible they become and the more affordable they become. So an example of how this works is if you were to take a clearing solution and put your sample in that clearing solution for several days, you would be relying on passive diffusion to get through the sample and clear out those lipids. So if, for example, on the far right, you see a brain that's still solid in the center, that's because the clearing solution cannot make it through to the center of that organ through passive diffusion alone. If you introduce an electric current with electrophoresis, you're actually able to remove most of the lipids throughout the entire organ, but you end up getting a deformation of your sample and you compromise the structural integrity of that. So something that they've developed is something called stochastic electrotransport, where they create a rotational electric field that essentially speeds up the process of passive diffusion. So for example, here at the bottom, we have a static electric field where there's no electronics, and the pink represents the solution going through the different molecules. So we don't have a lot of movement here. But if we introduce a rotational electric field, we can actually increase the rate of diffusion and we can get a much better clearing result. You can also use this with labeling. So for example, on the bottom, they just use passive diffusion, and we see that the center of each of those brains does not have very much contrast. But using stochastic electric transport, we see that we actually have a very even labeling distribution. And once you have that, you have your clearing, you have your labeling, you can put it onto the light sheet microscope and you can get that optical sectioning and essentially do a 3-dimensional reconstruction of your entire organ. And you can use that to trace different neurons. You could use that to look at structural compositions with disease and many other things. Something that's a little bit more possibly known to people is using something called 2-photon imaging. And so on the left, we have a traditional microscope where you have a cone of light with a focal point in the center. And then on the right, we have our 2-photon imaging, where we see that very, very small portion of that field is actually illuminated. And that's because it's a manipulation of how we deliver light to the sample. If you have 2 infrared photons of very, very low energy, they're able to travel through the tissue more effectively than high energy light. And the combination of the energy of those 2 photons at the same time is actually equivalent to an excitable photon in a shorter wavelength. So for example, if you wanted to excite a green fluorescent molecule, that's about 488 nanometers in green light, you could use 2 photons of infrared light to excite that one fluorophore. And one of the reasons that people do this is for something called intravital imaging. So if you wanted to look at how the lung or the brain or kidney dynamics were inside your mouse, but you don't want to remove the organ because then it has to be fixed and dead, you can do something called intravital imaging. And there's this particular lab here that has many different models on how to do intravital imaging in brain, in lung, in spleen. And while you do need specific equipment to do this kind of microscopy experiment, it does require a lot of optimization, but you have the benefit of having your samples live, and you can use endogenously expressed fluorescent proteins, or you can introduce contrast in other ways. So moving on from organs and going into tissues, this will probably be a little bit more familiar to people, where we have an example of a histology image of a mammary gland duct. So these mammary gland ducts were sectioned and they were stained with something called picocyrus red. And based on these two sections, we can't really tell that much about them. However, one of these has a structural defect and one does not. So we can actually take a really old technique called polarized light microscopy, and we can actually see a significant difference between the two of these sections. So essentially what happens is collagen is a fibril, and when it's fixed and stained, it actually takes on a crystalline structure. And that crystalline structure is able to manipulate light in a very specific way where it can generate this color. So this is not fluorescent light, and it's not color that's been superimposed. It's actually light that's been bent by the collagen fibrils to produce a different color. And this technique is actually really old, and it's not super ubiquitous anymore. A lot of people use it in things like geology. But essentially what you do is you put in a polarizer where you select wavelength of one specific orientation. And then you introduce another grate at the bottom called an analyzer, and you set it perpendicular to the first polarizer. And if you have a sample in there that is birefringent, which means that it's able to reorient the light in the center, you can actually capture all of the light that's been reoriented by the sample, and that reorientation of the light generates a different color. So what people did in this particular instance is they quantified all of the red pixels, the green pixels, the orange pixels, and so on, and they were able to infer that there were structural differences between their two different conditions. One of the other things that's very popular with tissue sectioning is something called confocal imaging. Confocal is incredibly popular because it's very accessible, it's affordable, and you get really good resolution. So for example, if I were to compare traditional wide field microscopy, which is essentially using just regular light delivered to the sample, no diffraction, with all the diffraction of the light included, and something called confocal, you can actually see that the wide field has significantly less resolution than the confocal image on the right. And the way that this works is very similar to a traditional microscope. However, you have the introduction of something called a pinhole. So you have a pinhole that helps focus the light that is being excited onto your sample, and you're able to illuminate a very specific part of your sample in X, Y, and Z. So you have again, a sample that has a green fluorescent protein, so it emits green fluorescence. And then that light is also gone through a pinhole. So instead of having a large section of your sample illuminated, you have a very specific section of your sample illuminated, and that's increased resolution. You can then, at that same depth, image the rest of your sample, and then you can change the depth, and you can image in those exact same places again. And by doing that, by imaging in X, Y in one plane, and then moving to the next plane, you can generate that three-dimensional representation of your sample. A subset of confocal imaging is called spectral imaging. Spectral imaging is really unique. It does require a specific type of equipment, but if you, for example, wanted to look at a breast cancer tumor section like this image, and you wanted to look at the immune cell infiltration. What you could do is you could use spectral imaging to label multiple things, and you could do a neighborhood analysis of what cell types are in specific areas. So the way that you do this is confocal imaging will typically only allow you to image up to 4 colors. The reason you can only image up to 4 colors is because once you go beyond that, there aren't enough lasers that are specific in between, and the colors start to overlap. So they start to bleed into each other. So if you have an image in green and an image in yellow, they're very close together. And it gets to a point where you can't actually differentiate one from the other, unless you have something called a spectral detector. So for example, here we have 7 different colors, all labeling different things. And we can tell that they're all distinctly different structures. We don't have to worry about them overlapping. And the reason is because when you do spectral imaging, you have to build something called a library. So if you have a tissue section with all 7 colors and you tried to image them, they would all bleed together. But if you have one section with 1 dye for each of your 7 dyes as your controls, then you're able to image that particular sample in increments of 10 nanometers. So we start at 421 nanometers, increase by 10, all the way through the entire visible light spectrum. We can actually select which wavelength is going to be specific to that dye. And we can do that for all 7 dyes, and that way we know that each one is going to be unique and specific to that fluorophore. Something else that's actually very interesting is precision live cuts. So instead of having a special light sheet microscope where you can cut out an entire organ and image the entire organ, you can actually use a vibrating microtome to section out live pieces of tissue. In this example, it's a section of lung tissue with immune cells going through the tissue and the airways and the blood vessels. So you don't need any special equipment. This video was taken on a regular confocal microscope, and it's compatible with routine labeling techniques. So there's no special labels that are needed. If you have live dyes, you can use that. If you have antibodies, you can use those. And you can image them through them, and you can get an idea of what's going on in your sample if you don't have access to special equipment. So now we'll move on to single cells and intracellular dynamics. Single cells are probably one of the most complex things to discuss because they've been around for so long and they're the easiest thing to image because of their size. So when we're talking about single cells, it is incredibly important to think about what kind of environment you're putting them in. You need to decide if you're going to do a 2-dimensional assay or a 3-dimensional assay, and that's going to depend on your type of experiment. If you're doing cell migration, you could do that in 2D or you could do it in 3D. If you wanted to look at an invasion assay of a, say, an organoid or a tumor microsphere, you would want to do that in 3D so that you can get a better physiological representation of what that might look like. Whether or not you do 2D or 3D might also depend on your cell type. If you have adherent cells, you can seed your cells into a regular plate and they'll migrate and they'll move and they'll be dynamic and they'll do what they're supposed to do because they're sticking to the bottom where you can image your sample. If you have non-adherent cells like immune cells, then they're not going to stick to the bottom. And if you're trying to image non-adherent cells, they're going to be moving as you're trying to image them, and you'll get a very blurry image. So the best thing that you could do is to embed them in a gel. And you can embed them in a biological gel, or you can embed them in something that is biologically inert, like a polysaccharide gel. But whatever you choose, it's very important that what you're choosing is something that's physiologically relevant. So, excuse me. For example, here I have collagen type 1 in the first column and the second column, but one is from bovine skin and one is from rat tail. And you can tell by looking at the images below, which is a reflection microscopy image of the collagen fibrils of these 2 different types. They're structurally very, very different, and they do have impacts on the phenotype of those cells. So we see on the bovine skin collagen cells that they're nice and elongated, whereas the rat tail collagen cells have lots of little projections everywhere. So if you have mouse cells, it would probably make the most sense for you to embed them in rat tail collagen because they're both rodents. But a lot of people use bovine collagen because it's so easily manipulated and easy to work with. Or if you have something like Geltrex on the right here, that is a heterogeneous gel that's supposed to mimic the basement membrane. And that particular gel doesn't have any fibrils. It has lots of ligands and protein fragments of laminin and collagen 4, but there's no actual fibrils for the cell to elongate itself on. So you actually get a very spherical phenotype. So whatever you choose, you should be looking into what type of matrix is most relevant for the type of experiment that you're going to be doing. Then you have to think about what you're going to label your cells with. Antibodies are really common. One of the downfalls of using antibodies is that means that your cells typically have to be fixed, which means that they have to be dead. So one of the things that you can do with antibodies is you can look at protein localization. Is your protein localized at the membrane? Is it in the nucleus? Does it translocate to the nucleus at specific time points following a drug treatment? These are all things that you can do where you fix your cells at specific time points and label them with antibodies and image them to quantify that. You can also image the cells to look at phenotypic changes following drug treatments or in specific conditions. Something else that is becoming very popular is using live stains or fluorescent proteins. So you can have endogenously expressed fluorescent proteins like GFP tagging your protein of interest, or you can have organelle stains like I have here. So for example, in green, we have a lysosomal stain. In red, we have a lipid stain. In purple, we have a mitochondrial stain. And then we have our nuclear stain. So if you wanted to look at intracellular dynamics, there are lots of proprietary dyes that you can use to label individual structures, and you can image them over time. If you're doing live imaging, something that you can do is you can dive into something called photo manipulation, where you have specific endogenously expressed fluorescent proteins or dyes that have been added, as long as they have these properties, and you can manipulate them using light to find out specific information. So for example, one of the most common types of photo manipulation, because it's the easiest to achieve, is called fluorescence recovery after photobleaching, or FRAP. And that pertains to the top row example here, where I have photobleaching, where you have a green fluorescent protein that has been photobleached. And when you photobleach a fluorescent protein, essentially what that means is that the fluorophore structure has been physically broken and it's no longer able to absorb that light and emit fluorescent light. So one of the reasons that you might do this is if you had your cell and you wanted to look at the trafficking of a specific protein. What you would do is you would take a very specific section of your cell and you would completely photobleach that section. And then you would measure how long it takes for the fluorescence to recover in that area that you completely photobleached. It can give you an idea of the organelle trafficking in there, or it can give you an example of the protein of interest that you're looking at. Something else that you can do is you can photoactivate proteins. So for example, here we have a non-fluorescent protein, but if that fluorescent protein was exposed to high levels of UV light, it would actually activate that protein and it would become fluorescently green. And it's another method of doing intracellular dynamics for mitochondrial fission or fusion, protein trafficking, and so on. A really interesting one is also photoconverting. I think this one in particular is very interesting. For example, we have a green fluorescent protein that would have been excited with UV light to convert it to a red fluorescent protein. And something that people will use that for is for things like lineage tracing, where you're studying development and you have a particular cell and you want to see where that cell migrates and where it differentiates. So you would be able to convert one green cell to a red cell and follow it and all of its daughter cells down the line through development to figure out where they go. One of the other things you can use it for is for structural changes in your sample. My previous supervisor, Luke McCaffrey, did an experiment with this where he was looking at mammary gland duct elongation. And he wanted to know if the ductal elongation was happening at the end, at the terminal end bud, or if it was happening within the body of the duct itself. So he actually just photoconverted strips along the duct and then imaged them later. And he actually found that the duct was elongating in the body of the duct and not at the terminal end bud. So he was using them as a marker for structural changes, which is very interesting. To give you an example of what that looks like, this is a video that I took of mitochondria, and this is a green fluorescent dye. And I imaged that green fluorescent dye through time. And then I took a section here and excited it with UV light at high energy. And I converted that one particular section to a red dye. And I would have been able to continue imaging that particular section over time to see where the mitochondria from that section ended up over time. If you're going to be doing live imaging, it's very important to consider the conditions of your cell, of your cells. So for example, you need to have the pH of your cell, of your media buffered, and you need to have it at a neutral pH if that's what your cells like. You need to maintain constant temperature. We think about culturing cells, they have to be at 37 degrees, and you have your humidity and your CO2. This is an example of a video where the media wasn't buffered and the cells were not given CO2. So every time that you see a green fluorescent cell disappear, that marks the death of the cell. So we have to make sure that the conditions of our imaging experiments are very well tailored. Something else that we need to take into consideration is the phototoxicity. It's not something people think about very often, but if you think of your cells and you think about the ones that are in your tissue, they're not exposed to light ever. So when you have an imaging experiment and you are exposing them to high amounts of very specific energy, they're going to generate reactive oxygen species, and it's going to be very detrimental to their health. So the way that we deliver that light to them is incredibly important. One of the things that we need to consider is the exposure time. In this example, we have cells that were imaged in a very long exposure time, really high power light. And every time that that green light disappears in the video, that's an example of a cell that is dying. In this particular example, we're looking at the power of the light source, not just the exposure time. So these cells are imaged continuously and they're imaged with very low power. So we can see that 20% power ends up bleaching the cells within 15 seconds, whereas a very low power is able to go on for about a full minute. So when you're setting up your experiments, even if it's live or it's fixed, it's very important to consider your exposure time so that you aren't purposefully photo bleaching your sample. And if they're live, that you're not accidentally killing your cells because you're trying to get a better image with a higher light source. Something else that you need to take into consideration is your fluorescent dye choice. I just have one example here regarding nuclear dyes. So on the left, sorry, we have a dye called HUSH, which is the live version of the DAPI stain that binds DNA. So if we look at this particular video, we see this cell dividing, and it actually snaps back. It can't divide properly, and that happens again. And those cells have become multinucleate. And one of the reasons for that is because that particular dye affects the DNA rigidity and elasticity. But you're also illuminating that dye with UV light, which is very high energy and very detrimental to the cells. It can actually induce DNA damage. So if you have something instead, like this DRAC5 dye, we can see the cells dividing and they're able to split and divide normally. And part of the reason is because we're using red light to excite, we're using red excitation light to give far red fluorescence, which is going to be much lower energy, much easier on the cells. But the other thing about this dye is that it doesn't affect the rigidity. Both of them bind the AT-rich regions of the DNA in the minor groove. So they can affect how the cell works, but if you have to use a nuclear dye in your live experiments, it's best to choose one that's going to be the most gentle on your cells, because you don't want to manipulate the behavior of your cells. So then we move into single molecules. So everything that I've talked about before is what we would consider to be conventional microscopy. We're using regular light, and we are removing some of the diffracted light to increase our resolution. But the majority of all of the techniques that I showed you thus far are what we call diffraction limited. They're able to achieve a 200 nanometer resolution in X and Y, and they're not able to go beyond that. So that ended up generating the need for something called super resolution microscopy. And I'm going to talk about 3 different kinds of super resolution microscopy in brief detail. The first step down is something called SIM, which achieves 100 nanometer resolution, and then something called STED, which is between 30 and 70 nanometer resolution, and then STORM, which is between 10 and 55 nanometers of resolution. So SIM stands for Structured Illumination Microscopy. And essentially what you do is you have a graded pattern, and you have that grate over your sample in a specific orientation at a specific angle, and you take an image. Then you move that grate to a different angle, and you take another image. And you do that multiple times, and each time you change that, that's called an iteration. So you do multiple iterations, and then you use algorithms to reconstruct the image after the fact. So on the right here, we have an example of a confocal image, and we can tell that there are actin filaments there, and we can tell that there are mitochondria there, but the resolution is a bit blurry. And the reason it's blurry is because it's diffraction limited. Like I said, we can only achieve 200 nanometer resolution. That light is out of focus. If we use SIM and we reconstruct that image, we can see increased resolution of those actin filaments and those mitochondria. Then if we move into something called STED, which is the next step down in resolution, or next step up rather in resolution, this is a particularly interesting one because it actually just manipulates the way that the light is received by your system. So for example, we have the central area, our diffraction limited area of illumination, and we have protein 1 and protein 2. And if we put those through the focal beam of the light, we can't actually tell the difference between those 2 proteins. And that's because they're too close together. We can't resolve them as separate objects. But what STED does is it creates this donut where the outer ring of the donut forces the fluorescent molecules to be non-fluorescent, and they only fluoresce in that central part of the donut, and we're able to resolve those as 2 distinctly different objects. So on the right here, I have an example of a confocal image of microtubules, and the exact same sample imaged with STED, where you can see that they're a lot more spotty, but that's because we're looking at individual proteins, and we can see that they have increased resolution. So the reason I'm talking about these is because we hear a lot about co-localization analysis, and if we look at a confocal image of protein A and protein B, if we were to look at that particular image and we did our Manders coefficient or our Pearson coefficient to determine how co-localized our 2 proteins are, we would say that they are co-localized. But if you were to put this into a super resolution microscopy image, you would actually see that they are in fact not co-localized. They are just very close together now that we can actually resolve them as 2 separate proteins. So if you want to do co-localization analysis, I would highly recommend that you try to find a super resolution platform. If you can't do a super resolution platform, at the minimum, you should be doing all of your images in 3 dimensions, sampling as much as you can, because when you have all of this diffracted light manipulating the size and location of where those proteins actually are in space, you need to try and accommodate for that as much as you can. The last type of super resolution microscopy that I'm going to talk about is called STORM, Stochastic Optical Resolution Microscopy. And this is one of the types of microscopy, super resolution microscopy, where you would need to have specific types of fluorophores in order to achieve this image. A lot of these techniques, you can use conventional fluorophores, Alexa fluor, probes are very popular. Many other types of dyes that are common are really popular. But with STORM, you need your fluorescent molecules to blink. And that essentially requires you to take multiple images over time so you can catch different fluorophores blinking at different times. And similar to the SIM application, we have multiple images taken over time and then reconstructed over time. So we have example again of microtubules because they're a perfect demonstration of how much resolution you can achieve, where you have conventional microscopy where you cannot tell that there are individual filaments there, versus STORM where you can see individual filaments. And someone actually very creatively represented this using the Eiffel Tower, where they took multiple images at different times to get the blinking lights on the Eiffel Tower, and then they used a particle detection algorithm and then overlapped all of those images together to get a fully reconstructed image. So I know I'm a little bit under time. There's a lot of material to go through. Unfortunately, it's impossible to cover all kinds of techniques that you can use on all the different samples and all the different things that you need to consider in time. So hopefully this presentation sparked a little bit of creativity in how you want to approach your next imaging experiment or how you want to diversify the work in your lab. And if you have any questions, I'd be happy to answer them.
Thank you very much, Rebecca, for the presentation. So yes, if there's any questions, please feel free to ask them, or you can put them in the chat box.
No questions? I don't know if anyone had an example that they wanted to go through of some specific problem that they had or not sure how to approach a particular experiment.
I guess I had a question. So if I have, because I'm working with human neutrophils, and I was just wondering, so, and they're non-adherent cells, so they're in suspension. So what would you recommend I use as the matrix, I guess, to put them in the gel?
Yeah, so it's interesting because I'm in an immunology lab now and I've never worked with non-adherent cells. So I'm currently in the exact same place as you and trying to kind of reimagine how I used to image things and how I need to image them now. And so it is important to embed them in a matrix. Neutrophils move very quickly, if I understand correctly. Yeah. And are you looking at their dynamics or you're looking at things intracellularly?
Their dynamics, so their migration, for example.
So if you're looking at their migration dynamics, you would want to choose something that's biologically relevant. I think that given their size, they're so incredibly small that you would probably be best using rat tail collagen. The reason I think that is because you can change the pore size based on the concentration of collagen. You can also change it based on the temperature that you polymerize your gel at. The bovine skin collagen, the pores are really, really big. And I think that they might even move like too quickly there. There's a whole thing about using matrices that are the same stiffness as your, as the physiology, as the environment that they would be in. So like, for example, in like lung tissue, I think healthy tissue is like 2000 Pascal or something like that. So you can have gels that are attenuated to the same stiffness and general pore size.
Okay. Thank you so much.
Yeah, no problem.
Are there any other questions? If not, thank you very much, Rebecca and everyone, for attending this seminar. And we look forward to seeing you in next month's seminar in April. And yes, thank you very much. And I wish you a great rest of your day.
Thanks for coming, everyone.
Bye.